This is a production of Cornell University.
Thank you, Jason, for the intro. Indeed, I'm the advisor of Judith, who, if you've seen her talk, works on meteorology, so to say the environment that the plants are exposed to. This is also my background. I'm actually trained as a hydrologist, work a lot across disciplines with meteorology. But like I always say, we have a special fondness for plants. And this is sort of how I ended up working on photosynthesis in a very cool environment, or a very hot environment, I should say, Yellowstone National Park. So I'll just tell you briefly how, as a hydrologist, I ended up studying photosynthesis. And this all came through lichens. Lichens is a very interesting species, right? They're a symbiosis between a fungus and an alga or a cyanobacteria. And what is really cool about lichens is that they can dry out completely, and then as soon as you hydrate them, they restart photosynthesis almost instantly. So I was actually doing a sabbatical a couple of years ago with a hydrology group, and then there was a group that invited me and said, hey, would you like to come and have a look at our work on lichens, because they have this very cool behavior in relation to water. So I ended up studying the water transport in lichens with them. And another thing we did is that we started looking at oxygen evolution from photosynthesis, sort of playing around. And then what we found out is when we expose the lichen to light, then dark, then light, then dark, we saw this oxygen evolution accelerate with subsequent light exposures. So we thought, you know, what could be happening there is that, of course, the alga produces oxygen, which is beneficial for the fungus, but then the fungus respires, produces CO2 that is then beneficial for photosynthesis by the alga. So we were able to prove that that actually was part of the symbiosis, in addition to the known symbiosis of water exchange and sugar exchange between the fungus and the alga. So that is, and not so surprising either, right? Because this is a confocal microscopy image that I took while at Rutgers University that was hosting me for this research. And you can see they're right in each other's space, right? So it makes sense that they're exposed to each other's gas emissions, so to speak. So this led us to become curious about the role of CO2 in photosynthesis, because the group I was visiting, Charles Dismux's lab at Rutgers University, they had mostly focused on the light reactions around photosystem 2, if you're familiar with photosynthesis. So this is where water is oxidized, or is split into oxygen and hydrogen protons. But then we said, so, you know, now that we saw this effect of CO2 on photosynthesis, it is sort of known, right, that with higher CO2, there is a fertilization effect where the gross primary production is increasing. We also know from greenhouses that I'm sure you're much more familiar with than me, is that when they enrich in CO2 up to about 1,000 ppm, this really benefits growth, right? It improves the fruit quality. And then, this is explained by the fact that higher CO2 levels enhance the saturation level of the rubisco enzyme that plays an important role, right, in carboxylation, so the CO2 uptake and then conversion to sugars. But there's also a couple of other interactions that are not so well understood. So it's not straightforward, as in, you know, more CO2, more primary production. And then just a bit of background of why CO2 is so important for photosynthesis, right? So here is this is a chloroplast, and then here is rubisco, this enzyme that fixes CO2. And then, through the Calvin cycle, so a series of reactions, it fixes CO2, converts it to a couple of intermediate compounds, eventually goes out as sugars. And then there is this ribulose bisphosphate that needs to be regenerated in order to take up the next CO2. But now what rubisco, this enzyme, also does, it can react with oxygen, the oxygenation reaction. And that is actually not beneficial for the plant. It costs energy. It produces CO2 rather than fixing CO2. It produces compounds that the plant needs to get rid of. So what you want is more carboxylation and less oxygenation. So you can imagine that by increasing the CO2 level, it is just more likely to pick up a CO2 rather than oxygen. right? So that's a bit of the background. And then the other thing that I'll be coming back to is that in this series of reactions, the Calvin cycle needs ATP and NADPH to run the Calvin cycle in these reactions. And these are produced by what's called the light reactions in the thylakoid membrane. So they capture the light energy that can be used to produce these ATP and NADPH. So they're very connected. And that'll be important as we'll see later. All right, so there's a benefit, right, of having higher CO2 levels. But on the other hand, very high CO2 can also lead to acidification of the cell environment. Because CO2, it's a tiny molecule, right? So it can easily permeate through membranes. It hydrolyzes, and then it forms acids, the Lewis acids that are just the hydrolyzed CO2, and then the Bronsted acids, so they actually release protons into the environment. And then what that does is that it reduces pH gradients, and these pH gradients across the membrane are needed to produce ATP. So you can imagine that when the cell environment becomes very acidic, it becomes harder and harder to build up a gradient across the membrane because there's already so much acid in the environment. So that makes it much harder to produce ATP. So there, a very high CO2 level can actually become harmful. Plus, it can carbamylate the proteins, so it can cause further damage. So we were wondering, so if we have elevated CO2, how does photosynthesis adapt, right? How does it make sure to take the maximum benefit from higher CO2 versus avoid this acidification effects? And we wanted to look at extreme CO2 environments. Most of the research has been done on sort of mild CO2 enrichments up to the 1000 ppm level. But we wanted to see, what if we go beyond, right? Where is there somewhere a threshold where it flipped from being beneficial to being very harmful? So this is how we started looking for an environment where we could find this out in the field. Because you can imagine that doing this, I mean, in the growth chamber, is that very representative for the real reality? Doing it in the fields, this is being done. They're called the FACE experiments, FACE. But there they find out it's hard, right? You blow extra CO2 into the field, but to get that well distributed and everything. So we wanted to look for a place where this is just happening naturally. And then, sort of a natural laboratory where this happens naturally is Yellowstone National Park. You're probably familiar with it, right? It's a volcanic area with a lot of geothermal sources. There's geysers, there's hot pots, there's several of those are emitting very high levels of gaseous CO2. And have been doing so for many centuries. Based on the geological research history, that is likely. And based on records from the USGS, we know that at least for the last, say, since the 1980s or so, they have records where they have recorded these CO2 levels. So we have a pretty good idea of also the history of CO2 emissions around these geothermal sources. So we went in 2019, and we went to scope out sites across the Yellowstone National Park that had been reported by USGS as having very high CO2 emissions. And very high, we're talking up to about 6,000 ppm, or even beyond in some places. But of course, we wanted to study photosynthesis. So we also need places where we find plants, right? Where we find life, or lichens. We also look for lichens, but we didn't find those. It's too harsh an environment for the lichens, apparently. They don't like an acid environment. But we did find vegetation around some of these sources, as you can see. So that was successful in that sense, that we could really find a couple of geothermal sites, or CO2 emission sites, I should say, with vegetation close by. But then, we found these two sites to be the most stable in CO2 emission, extreme CO2 emission, this 6,000 ppm elevation, more or less. And then also vegetation growing very close to the sources. So we could be pretty sure that they are really exposed to that very high CO2 level. They're both calcium deposits, calcium carbonate deposits, and that is what causes these very high CO2 levels as the gases move through the soil. You can imagine that takes up all the CO2 and then emits it into the atmosphere. Now, the other thing we needed is sample species that would grow very close to those sources, but also in a controlled environment, as in ambient CO2, 400 ppm more or less, so that we could compare between the two. So, for instance, this is panic grass that you see on the photo. Oh, I'll point it here for the Zoom attendance. So, panic grass is famous for growing close to these sources in Yellowstone. It has adapted to that environment, and it only grows there. So, for us, that was the first species we thought of targeting, but it turned out not to be useful because we had no control. We had no comparison to an ambient. But then Prunella vulgaris, which is actually a very common plant, it grows in lots of environments, including close to the CO2 sources, as well as in a more ambient environment, so 400 ppm CO2. So this is a species we could really compare between the high and the ambient CO2 environments. Another attribute of Prunella that was very useful for our work is that it doesn't like to close its stomata. In the sense that, you know, even when we harvested samples from the field, took them to the lab for experiments, their stomata would still be open. And this is important if we want to study gas exchange, obviously. So had they closed their stomata, there would be another no-go. But so Prunella was very helpful in that regard, that we could really study photosynthesis effects even when we had taken them from the field into the lab. So we wanted to study the effects of CO2 levels on photosynthesis. So this is basically what we wanted to study, right? Close to this Rubisco enzyme that fixes CO2. But to measure this directly, it's kind of hard. I mean, you can measure gas exchange, but then there's a lot of, well, first of all, it's an indirect measurement because you measure a net CO2 uptake, but where the CO2 goes and how fast it reaches Rubisco, you don't really know. Plus, it takes a lot of time. And we wanted to look at dark to light induction of photosynthesis, which is a dynamic that we needed to have more high resolution, time resolution measurements. So instead, we used a method that's called chlorophyll variable fluorescence. And this is fluorescence that's emitted from photosystem 2, so from the light reactions that produce ATP and NADPH for the Calvin cycle, as you can see here, sort of in a simplified scheme of photosynthesis. So the way this works is that photosystem 2 absorbs the light energy, and then it gets excited, and then charge separation happens, and the electron can be transferred to its primary electron acceptor, QA, called QA. And then from QA, once it's reduced, it can then again transfer the electron to PQ, a plastoquinone that then is converted into plastoquinol. And then from there on, in a series of reactions, eventually NADPH, NADP plus is reduced to NADPH. And by pumping protons across the thylakoid membrane at the same time, we produce a proton gradient that is used to produce ATP. So that's the connection. But now, once this has happened, so photosystem 2 has gotten excited, it has transferred its electron, there's only one acceptor. So at that point, we say PS2 is closed. It cannot take another electron, right? Because it's closed. The carrier already has one. Now, if the pigment gets excited, the energy cannot be transferred through the electron. It gets emitted as fluorescence. So that means that as long as photochemistry can be done, the electron can be transferred, we have no fluorescence. But if as soon as the system is closed, we get fluorescence. So we have sort of a probe of the photosynthesis activity by measuring fluorescence. And there's also a little bit of energy that can be released as heat. Yeah, I'll go into that later. We have a measurement technique that really we can tease out the variable portion of fluorescence, and that is not so sensitive to the portion that goes into heat. But that is an issue when interpreting this kind of observations. So that's what we're going to do. We're going to monitor fluorescence emission from photosystem 2 as a probe for what is happening in this chain of reactions that eventually produces the compounds that run the Calvin cycle. And thereby we can say something about whether or not CO2 is being reduced, taken up to form sugars. All right, so then another thing I need to say, in case people are familiar with the fluorescence method, the method that's called PAM fluorometry. I don't know if you're familiar with this. This has been used a lot in photosynthesis research. This technique is slightly different in that it's called fast repetition rate fluorometry. It uses ultra short, high intensity light pulses to excite the chlorophyll pigment, photosystem 2, and then measure, as you can see on the bottom, so it measures the chlorophyll fluorescence that rises as you shoot light on the pigment. And then as the photochemistry goes down, because the system gets closed, right, this QA gets reduced, we see the rise in fluorescence. Then we flash at a flash rate of 100 Hertz. So we have this ultra short pulse. We measure variable fluorescence. We wait about 10 milliseconds then, right, at 100 Hertz flash rate frequency. Then we get a little bit of reopening of the system, right? Some of the QA gets reoxidized in that time frame. So fluorescence goes down again. We flash again, fluorescence goes up, but the variable portion is much smaller now, right? Because the system is still partially closed. So we continue to flash and we monitor variable fluorescence. So this is typically what this looks like for 50 flashes in a row. And this is the variable portion of fluorescence normalized by the maximum of the fluorescence at that particular flash moment. So the maximum FM right here. And so what we see happening is that as we continue to flash, it becomes harder and harder to reoxidize between flashes. So in the end, the variable portion goes almost down to zero, meaning the system's completely closed, right? We can't reopen it between flashes anymore. So QA got reduced, the PQ pool, if you remember, got completely reduced. The system is stuck, right, at that point. It can't do further photochemistry. But then we continue to flash. So, oh yeah, and another thing I should explain is we see sort of the oscillations here at the start of the flash train. So this is a dark adapted sample. And what these oscillations represent is the oxygen evolution from the water oxidation complex. This happens in 4 steps, and that is why you see these oscillations. So that's another interesting feature of this technique, that you can actually measure the oxygen evolution from photosystem 2. All right, so now we do not one flash train, but we continue to flash. In total, we flash 180 trains of 50 flashes each. So the first train looks like what I just showed, right? So we started with an open signal system, dark adapted. We start to flash, we close the system, and then we wait a half second. Again, the system partially reopens. We flash again. We wait a half second, flash again. So we see a series of trains. We see the system reopening, closing, reopening, closing. So we're probing the reduction of the system by flashing, by these light flashes. And then what we hope to see eventually is that the system will reopen once the electrons are used to produce NADPH. NADPH is going to be used in the Kelvin cycle, so it gets reoxidized, and that means it can receive more electrons, right? So eventually you should see this shift of the electrons being moved into the Kelvin cycle. And then we can again reoxidize between trains. We expect the system to rise again. All right, so let's see if that happens. So this is the result, variable fluorescence over 180 trains, 9,000 flashes in total. So the start of the very first flash is this dark adapted variable fluorescence value, also called the maximum quantum yield of PS2, right? That's what people who have used fluorometry you're familiar with. Then we see the system closing, right? It goes all the way down like we saw, but now it's just squeezed. But then indeed, we do see the system reopening, right? We see sort of the overall levels rising a little bit, but then they fall. So the system starts to open, but then closes again. And then eventually, after about a minute, we see an overall rise of the fluorescence levels. And this is then the signal for us to observe the reopening of the system by the downstream reactions in the Kelvin cycle. So this portion in particular, we expect to be very sensitive to CO2, right? Because that's the Rubisco enzyme activity. Then the other thing is we see this very wide sort of comb width, right? So this is the difference between the start of the train. So we wait a half second between the flash trains. So we reopen the system partially, and then we fill it again. So this is sort of a measure of how much electrons come in and are taken out during the flash train. So this is the overall monitoring that we're going to do with samples from Yellowstone in the ambient environment as well as the extreme CO2 environment. All right. Then, yeah, so this sort of summarizes this, what I was just saying, that what we are monitoring with this, right? In particular, the rise in variable fluorescence, which is for us a signal of the Kelvin cycle activity. All right, and then the reduction, the comb width that shows us the ratio sort of between the light reactions that push the electrons in and the utilization in dark reactions that use NADPH and therefore reopen the system. All right, now let's see what happens when so this is still an ambient sample, right? So it's just used to 400 ppm CO2, but now we're exposing it to 6,000 ppm as the samples we found in Yellowstone. So this sample clearly responds, and as we expected, especially this last phase associated with the uptake of NADPH, or the utilization in the Kelvin cycle, responds very strongly, right? We see all the levels rising, so the system stays much more open. The electrons just flow through much faster. And also, even between trains, we don't fully close the system anymore, right? Because the electrons can really be pumped forward very fast. So this is sort of what we expected as a very strong effect of CO2. We also see that the initial levels are rising, right? So even very early on, electrons are already being taken out of the system and can be used downstream. All right, so strong acceleration of the reactions in the electron transport chain. Faster rise, faster overall levels. All right, now we're going to do the same thing, but we're going to look at plants from Yellowstone National Park that have grown in this extremely high CO2 environment. So they were always exposed to 6,000 ppm. Now, the first thing we see when we just look at the plant samples is that they're not doing great in the high CO2 environment. We see strong signs of chlorosis. We see the leaf sizes that are smaller. Yeah, the overall plant just looks very unhappy. And now we're going to do the same experiments looking at the fluorescent signal from these plants. So first we take the adapted plant. I'm going to call it adapted plant, so that has grown in 6,000 ppm CO2. And we're going to give it just 400 ppm CO2 and see what happens. And basically what we see is, well, sort of the start of the experiment. So we start out with a dark adapted sample. Then we start filling, reducing QA. We start filling the PQ pool, gets reduced. That sort of looks the same, right? The signals go down, they come up a little bit, but then they close again. But in this second phase that we associate with the Kelvin cycle activity, we basically see nothing happening, right? The signal stays down. So it looks like it's not activating the Kelvin cycle. It's not able to take the electrons out of the system. So the system stays closed all the time. All right, now let's see what happens if we give it 6,000 ppm CO2, right? Like what it's used to. And boom, we see more or less the same response as in the ambient sample. So as soon as we give it plenty of CO2, it can fully restore the Kelvin cycle activity. So the overall levels reached are, as you can see, this more or less the same. And also we see this much narrower comb width. So also between the trains, right, it can really very fast, it is able to reoxidize the system each time. The only slight difference that we see is that we see a sort of a downgoing slope here towards the end of the experiment. So there is some kind of downstream limitation here that starts to play a role, which we attribute to, if you remember the Kelvin cycle, we not only need to fix CO2, but for that we use ribulose bisphosphate that needs to be regenerated through the cycle in order to take up the next CO2 molecule. So we think that is a limitation in that downstream step for regenerating ribulose bisphosphate, because you can accelerate the carboxylation reaction, but you need the rest of the cycle to follow, right? So that's what could be going on here. All right, now this is a slide that summarizes the results. So far I've been showing results for single samples, right? So the box plots summarize data for the replicates. So we have 3 to 6 replicates for each of these groups. So we have an adapted group and we have a control group exposed to 400 ppm, exposed to 6,000 ppm, just like I was just the observation I was just showing. And I'm just going to summarize the results. So first, when we look at phase 1, right? So the start of the experiment, a dark adapted sample, we look at what happens, the maximum quantum yield of PS2. We basically see no significant difference between the box plots, right? So meaning that there's no significant difference between control and adapted plants. CO2 doesn't have an effect. Then when we look at the PQ pool size, so the next step, right, we reduce QA and then we reduce the PQ pool. Same thing, right? So this is where the signal goes down in the first train. That difference is also more or less the same, no significant difference between the groups. Then when we look at what we've called phase 2, so we saw that the signal going down again, right? So there is an intermediate downstream bottleneck where what we think is happening, the full chain gets reduced, right? So NADPH gets formed and waiting to be utilized by the Kelvin cycle. So there we get a sort of intermediate stop or slowing down. Again, we see that happening more or less similarly for all the groups. No clear CO2 effect, no clear effect between the different sample groups. But then for phase 3, we clearly see, as we saw in this, if you remember, there was this very wide cone width and the levels weren't rising, right? So much lower Kelvin cycle activity for the adapted plant in ambient CO2, but it's restored in higher CO2. The same thing for the cone width, right? So the reoxidation between the trains. Yeah, we also, here we see a clear effect for the adapted plants compared to the control. A clear CO2 effect to start with, and somewhat different between the control and the adapted plant. And then the other thing I mentioned is this downstream limitation by ribulose bisphosphate. We also sort of see that starting to appear mainly for the adapted samples and mainly exposed to high CO2. All right, so now how we see an adaptation effect, right, associated with the Kelvin cycle activity, but how can we explain it, right? What exactly can be behind this difference? Now for that, we're going to look at the various steps in the photosynthetic electron transport chain. So the way we often think about it is sort of as a linear response, right? So we have more light, more NADPH, ATP being produced, more CO2 uptake, right? Sort of the electrons flow through linearly, and then the products are used by the Kelvin cycle. But when you do this, then the ratio of ATP over NADPH that needs to be 3:2 is not correct. You don't get the right ratio. So we need to produce some extra ATP in order to be able to run the Kelvin cycle. So the sort of solution that is invented by nature to do this is called cyclic electron flow. So electrons pass through the system, reach a photosystem from photosystem 2 to photosystem 1, and then the electron is not transferred to reduce NADP plus to NADPH, but instead is cycled back, thereby pumping an extra proton that can then be used to produce more ATP. So it's all about tuning the linear versus cyclic reactions. Because if you would only do cyclic mode, obviously you're not going to have enough NADPH. So that is the trick then, right? For the plants is to make sure that these two systems are tuned correctly. And what we think is going on, and another thing I should say is that this has been shown also in the literature, is that when plants are dark adapted, they start out in cyclic electron flow mode. So they start out in this mode. It sort of makes sense that at the start, the first thing they start to do is to produce ATP, because ATP can be used for a lot of purposes, right? And it's beneficial to do that. But then as soon as the system sort of senses, I also need NADPH, it then turns on the linear electron flow, right? So you get a transition until you get the right ratio. So how we interpret our data is that because we see this adapted plant and we see it's not activating the Kelvin cycle. So it's very likely that it is in cyclic mode, but when it doesn't get a lot of CO2, it doesn't see sort of a reason, right, to kick on linear flow because there is a very low Kelvin cycle activity. It stays in cyclic mode. And that could explain why we don't see a rise in fluorescence. But then as soon as we give it 6,000 ppm, it does start Kelvin cycle activity, it starts needing NADPH, and then we do see the rise in fluorescence. We see the system opening up. So yeah, to summarize that, so what we've seen for the CO2 adapted plants, Prunella plants in Yellowstone, when we expose it to 400 ppm, so much lower than its growth environment, we see that the Kelvin cycle basically stays inactive. And we interpret this as, in this dark to light induction that we're looking at, the system, the electron transport chain, stays in cyclic electron flow mode. So there's very little linear flow. So there's very little NADPH available for the Kelvin cycle to do carboxylation. And the advantage of that would be, for a plant that grows in a stressful, high CO2, but also highly acidifying environment, is that it is always ready to make a lot of ATP, which it can use then for repair and homeostasis. So that could be an explanation. But then when we give it 6,000 ppm, the Kelvin cycle does get active to the same level even as the control plant. So this means that linear electron flow must be activated now, right? As soon as there's a lot of CO2. And then we get the right ratio in order to be able to run the Kelvin cycle. Now this is sort of confirmed by a next set of experiments that we did. So we had our 3-minute experiment, 180 trains, but we repeated it sort of in a light fluctuation experiment. So we have 3 minutes of light, and then we have 4 minutes. This is just practical because we have to change the system. 4 minutes of dark, 3 minutes of light, 4 minutes of dark. And then also we start out, this example, I mean, we did lots of combinations, but in this example, we start out with ambient exposure, 400 ppm, 2 sequences, then we elevate CO2 to 6,000 ppm, and we do another 2 sequences. And now if you look at this with having in mind that we start out with cyclic flow, so we start out with a system that closes very rapidly, and then it opens up in the upper level is the control plant, right? So it opens up, meaning it transitions to more linear electron flow, so that it can do so that it can do carboxylation. And this does not happen in the adapted plant. Now, if we wait 4 minutes in the dark, we sort of get part restoration of this cyclic electron flow mode. But when you look at the control plant, it's able to start up Kelvin cycle activity very fast, right? We see the minimum remains much higher. So meaning the system is already active. It's able to stay much more open. But this doesn't happen in the control plant. Not in ambient, which we didn't expect, but even when we give it a lot of CO2, we see the control plant being able to stay almost completely open, right? We see it hardly going down, so the system doesn't get reduced. It can reopen straight away, meaning that directly it establishes this balance, right, between cyclic and linear electron flow. Not so for the adapted plant, right? If we give it a dark exposure for minutes, we see the levels going down. It closes again from the start of the experiment, and it needs time. It does open with 6,000 ppm, but it takes much longer for it to activate. And we do it again, and we see the same thing. What we also see is this negative slope starts to appear, right? As we continue to expose to light, this downstream limitation starts to become more important. Is what it looks like based on this data. So this sort of fits the picture, right, with the 2 experiments. So then to summarize our conclusions for the Prunella plant, yeah, I'll just show them here. 400 ppm, we saw no activity on the Kelvin cycle. What we think is caused by the system staying in cyclic electron flow mode. 6,000 ppm, we saw no evidence of effect on the light reactions, right, on the PS2 light reactions. What we see is a transition from linear to cyclic. Oh, this should be the other way around, I'm very sorry. From cyclic to linear flow, when we go from dark to light, is much slower, right? Because it's closed in between, and only then could it activate. But then it becomes almost as active as more or less as active as the control plant eventually, which also tells us that there is not really an adaptation in functional Robisco content, because this has been an explanation, a possible explanation for adaptation to higher CO2. You would adjust your Robisco content, right? Because it doesn't work. It needs to work as hard, and it has a lot more CO2 available. But because it restores to the same level as the control, we think that is probably not what is happening in our adapted samples here. So that's also interesting to keep in mind. All right, that brings me to the end of my talk. I'd just like to thank a few people. First of all, of course, Professor Charles Desmukes from Rutgers University, the Waxman Institute, who introduced me to photosynthesis and with whom I've been writing proposals to get us to Yellowstone for these very interesting experiments, which have been supported by, of course, the National Park Service, who provided the research permit, the Waxman Institute, who provided a seed grant to cover some of our travel costs, and the 4TU Federation in the Netherlands, who provided some of the instrumentation we have been using in the field. So with that, I thank you for listening.
Questions? Oh, we've got a couple.
Mm-hmm.
Go ahead, Bill. How widely dispersed is this elevated CO2?
Around the high CO2 sources, you
Yeah.
mean? Yeah.
Meter scale, 100
Yeah,
meter
meter scale.
So
Yeah,
it's very, very localized.
It is. Yeah. As soon as you're, say, more than 10 meters away, in most cases, the levels are back to, also because of the air mixing, you're back to ambient CO2. Yeah, which is convenient because we're quite certain that other than CO2, the plants are exposed to the same environment, same soils, everything else. Yeah.
Yeah, so I was wondering, sometimes we see that when plants are in those conditions, right, they're exposed to extremely high CO2, maybe not necessarily adapted, but if you were to have a plant that was, synthetically, or just that, yeah, in a plant that has a very high CO2 environment, there's triose phosphate utilization limitation that occurs, and, that could also limit the rate of photosynthesis. Like, do you hypothesize that is some role in what you're seeing?
Yeah, good point. Yeah, this is another downstream limitation that could explain what we're seeing. Yeah. Yeah, that's a good point, especially at very high CO2. So we have a high production of sugars, and if they're not moving out of the system fast enough, then that also inhibits the ribulose bisphosphate regeneration. So yeah.
Okay.
Yeah.
Yeah, a colleague and I have been sort of thinking about doing some work at Yellowstone, also with high CO2. One of the things that we've been spending some time thinking about is that these emissions are not just CO2. There are other gases that are in there that also can affect the soil chemistry. Your adapted plant didn't look very healthy. And I was wondering whether you had any concerns about these other stresses that the plant is under in interpreting any of your research.
Yeah, that's a very good point. Yeah, I should have mentioned that we selected these sites because they're almost exclusively emitting CO2 and very low in sulfuric gases, other types of gases, also relatively low in water vapor, because some of the sources emit 90% water vapor and then a tiny bit of CO2. So they were really selected, the travertine deposits. So they really emit mostly CO2 as a gas and very low in other gas compounds that could be confusing our signal. Yeah.
Did you do the monitoring of the emissions or did parts?
USGS, yeah. This is the USGS.
Yeah, okay.
Yeah, we're taking from their reports.
Okay.
Yeah.
Did you studied any light intensity based cycles which could be different in these two different environments?
Here we always use the same light schemes for the fluorescence measurements. Yeah, so we haven't played around. That's another thing we could do, indeed, is play around with different light intensities and see what happens.
Based on our data, it's very clear there is sort of acclimation or adaptation. But I think your data, I think all the conclusions are dependent on the fluorescence data. A couple of things. First, when you compare chlorophyll fluorescence at 400 ppm versus 6,000 ppm, in the first case, you have photorespiration. And the second, at 6,000 ppm, photorespiration probably is completely inhibited. So the total electron transport measured by chlorophyll fluorescence, so represent, you have these two sinks, right? One is carbon dioxide reduction, the other is oxygenation.
Mhm.
So how do you differentiate those two? That's the first question. The second question is, you suggested that Rubisco, the amount of Rubisco didn't change,
Mhm.
but you never measured Rubisco.
No, we did not.
Yeah, so I really don't feel comfortable with that conclusion because without directly measuring Rubisco, the total amount, it'll be very difficult to make the conclusion that Rubisco, the amount didn't change. Because chlorofluorescence is an indirect measurement. What you're measuring is measuring the light reactions. So not a direct measurement of Rubisco mediated dark reactions.
No, but that's exactly why I put functional Rubisco to start with, because we didn't measure Rubisco content.
Okay.
But functional Rubisco, because if the functional Rubisco content would be very different, the samples would not be reaching the same variable fluorescence levels that we're seeing in the experiments, right? There would be differences when they're exposed to the same levels of CO2. So that's why, we're pretty confident that this is the case.
Yeah. But there's a way to directly measure the functional Rubisco portion.
Mhm.
To measure the initial activity that represents the amount of Rubisco that is actively engaged in photosynthesis. And they can measure the total amount of Rubisco. That's the total amount. So that's the ratio is called activation, percentage of activation. You could directly measure the initial activity of Rubisco to support this conclusion, I think.
Right.
That would be much better.
Yeah, that would be very interesting. The thing is, of course, here we are in Yellowstone National Park with very limited facilities. So we build up sort of an improvised lab to do these experiments, but it's not easy to do every kind of measurement that would be ideal, to confirm all the conclusions. What we do plan to do is to go back with an instrument that can also measure the oxidation state of P700, so photosystem 1, and also to measure the proton gradients, which you can all do with optical measurements. So they're relatively feasible for this environment. So that would at least, with that we could corroborate some of these conclusions. Because I agree, it's only one method.
Yeah.
I fully agree. But yeah, so we hope to fill some of the holes with these additional measurements in the field. Yeah, Laura?
Hi, I'm going to talk to you later in more detail about this. I kind of agree it'd be really great if we could sort of reject the hypothesis that this goes involved. And understanding the field limitations there, of course. I have a very naive question about what you're measuring with the fluorescence. Is it Rubisco activity or is it essentially like IUPDP reduction? Is it the regeneration phase that's being measured?
What we measure is a net rate of uptake of electrons.
So yes, so it's when the NAD, et cetera, is being, so that's in like when it's going from like 3PGA to G3P, right? So it's measuring the rate of IUDP regeneration essentially, which is consistent with when you're seeing like that downshift, and that's becoming limiting. It's just not in my head whether that is a proxy for Rubisco activity or not. But this could be a fun discussion.
Maybe the terminology, you're right. Maybe, correct my terminology if I'm wrong. But what we really measure is the utilization of NAD, or the reoxidation of NADPH by the Kelvin cycle, because we see strong CO2 effect sort of confirms that it has to do with, but I shouldn't call it Rubisco activity because that is not what we measure. You're right. I mean, the correct term is reoxidation of the electron transport chain. Yeah.
How much of the inability of the adapted plants to efficiently use the ambient levels of CO2 do you think is related to the chlorosis and the general lack of health of the plants? That it would just be harder in general for it to photosynthesize?
It depends. So if it's in its high CO2 environment, it's always exposed to 6,000 ppm. It would be able to do photosynthesis, right? So then that would not be causing the chlorosis that we see. If, on the other hand, the CO2 levels are fluctuating because of air mixing, then that would definitely be part of the stresses, right? Because then it would be exposed sometimes to this very nice and high level, but then it goes down, it goes up, it goes down. So then that would be part of the explanation. Yeah.
I have two questions. One is on that part right there. So when you take the adapted plants and they're at the lower ppm, that's where you see the cyclic kick in, right? And you're generating too much ATP relative to NADP. Where does that ATP go if it doesn't go? Where does it get shunted if it doesn't get used?
Yeah, well, this is something I think a lot of you people know more about than me. But ATP is used for a lot of processes in living cells as an energy carrier. So
Yeah,
how it gets shuttled out, I'm not sure.
Yeah, so because I was thinking maybe that's part of the toxicity, right? If it's getting shunted into something that is hurting the plant, then
The ATP itself, you mean?
Yeah.
Could be.
But see,
Okay.
did some other thing. But then the other thing I wanted to ask you is about, so you have this swath of the plant that's getting the high dosage, and then the adjacent swath is the control.
Mm-hmm.
So if you take the high adapt and you maybe, I doubt you've done this, but maybe, and you put it in a 400 ppm and just keep it there forever, does it ever acclimate back to 400?
Yeah, that's a very interesting question. Yeah, yeah. For that, I mean, so it would be hard to do in the field. I mean, we would need to be sure to be able to come back every year, and it has a very short season because we're at more than 2,000 feet, 6,000 feet altitude. But this is something we're thinking about. If we can bring the plants out of the, it is included in our permit now that we can bring plants back from the field. And then we could see if in the greenhouse or in growth chambers, we expose them to 400 ppm and whether they react. That would be very interesting, right? To see if it's elastic.
Yeah, and then also just to move the 400 into 600 until
Right.
you see a decline, right? Because the first part of the hypothesis was that CO2 can be detrimental at high, right? And so then to like model that breakdown, basically.
Yeah, yeah,
Okay.
yeah. Yeah, that's something we need to find funding for though, because that's going to be an extended set of experiments, but yeah.
I'm thinking back about the plants again. Are there plants that are growing in
Yes.
a high CO2 environment that actually look healthy? Because the ones that you did show, they look pretty ragged, and I'm trying to imagine the leaves that you would look at to do these experiments. Did they have healthy leaves, any of these plants?
They're a lot smaller, but some of the leaves do. I mean, this in the kind of measurement we do, we need only a small portion of the leaf to be healthy to do our measurements.
Okay, so because the one, I don't think, okay.
Yeah, no, that was a pretty crappy example. You're right.
Just before we go back to the crowd, we have one quick one here.
Yeah.
You might want to just say it out loud.
Yeah, there's a question from the Zoom audience. Did you notice any differences in stomatal conductance at different CO2 concentrations? Yeah, so this is sort of the nice thing about Prunella is that it didn't respond very strongly in terms of stomatal conductance. So we took stomatal conductance measurements in the field and we brought samples to the lab, we measured in the lab, and we didn't see strong differences. I mean, stomatal conductance, there's a lot of variability to start with, right, when you measure it in the field. But that degree of variability and range didn't change for samples, control or adapted. And even when we brought them to the lab, the conductance got smaller, but the stomata did stay open and also to a similar degree, more or less, right? Within the variability range that is inherent to the measurements.
You sort of mentioned this before, but I just want to ask again, how far apart were the exposed plants and the control plants?
Order of magnitude 10 meters.
Excuse me?
10 meters, more or less. So that is 10, 12 yards or 30 feet.
30 feet. That's about 30 feet.
Do you think your control plants are really control plants? I mean, the wind is blowing this stuff every which direction. Okay. So if they're only 30 feet apart, my guess is that at some point your control plants were being exposed to elevated CO2 also.
It's rather the other way around, I think, because it's a point source of a very high CO2.
Yes.
So plants growing very close to that point source do get the 6,000 ppm, but
To the outlet where the stuff came up out of where the CO2.
within a foot.
Okay.
They grow directly at the edge of the source. Yeah, and the soil is not hot, so these are not hot sources. They're really CO2 sources.
Okay.
Yeah.
Is there like, so 6,000, is that the extreme that was identified, or that's like consistent, like consistently around 6,000 around these hot sources? For the.
This is the level that we measured out in the field at about 6,000 ppm.
Okay.
There's been some sites where they found up to 10,000 in the USGS reports, but those sites had no growth, no vegetative growth around them. So.
Great. I was just wondering, like, if you were to measure with, and I'm not sure if this is like part of your design or not, but if you measured at sites that had lower, like 2,000 to 3,000 ppm, right? So like some of the gas exchange systems can ramp up CO2 to like about 3,000, right? I mean, if it's like you compromise some of the, the extremity, I guess, of the CO2 level, but at the same time, you'd be able to actually like get net CO2 assimilation and get the direct parameters. Like, do you think that'd be something you'd be interested in doing in the future?
Well, this is actually what we intended to do when we went. We thought about measuring along a transect with over a range of CO2 concentrations. But this turned out to be hard. Exactly for that reason, it's very hard to get something of a stable CO2 concentration transient in the field with all the other variations. So we stuck to very high, very close to the source, and then ambient far away enough from the source that we had a stable ambient. What we did do, I can show you that data. In the lab, because Prunella is a common plant, right? So you can actually just order it and grow it in the lab. And then we did, here we did the range of CO2 experiments where we start out with an ambient signal. And then here we expose it to just 0 CO2. So you see this last phase, basically, system staying closed. And then we had a couple of intermediate levels, so 1,600, 3,000, and 6,000 ppm. So these are lab-grown samples now. So there we were able to control a lot more things. So they're grown in ambient CO2, and we looked at the CO2 effect. Just to get an idea of what it looks like, right? What the intermediate levels look like.
Okay. So sort of connected to question about like elasticity, but when you saw the difference between the control and the adapted samples, do you have an idea of like how much of the difference you thought was like due to like actual like evolutionary adaptation, and how much was due to the like acclimation of just being raised in this environment?
Yeah, that's a good question. It would be great if we could do all the genetic analysis, but we haven't done that. We didn't bring samples from the field in the times we went. But so the reacclimation experiment would be one to do to see if we can confirm acclimation versus adaptation. Probably easier even than the genetics, because I don't know how well the genome of Prunella is known, but that might be very hard to trace back. But this is something that would be very cool to do, to confirm which of the two.
Let's do one more and then I also want to say there's the student lunch in Man 413 just down the hall. So you had your hand up a couple times there. Let's do that and then close it out.
Did you notice any like changes in density of the Prunella as you approached the CO2 source as compared to more of the control populations?
Can you speak up a little bit? I just couldn't hear you.
Any changes in the density of the Prunella as you approach the CO2 source as compared to the control?
The density, you're saying?
Or abundance?
That's kind of hard to say. There's so few plants growing very close to these sources. We're going back this summer, so I'll keep an eye out and see if that is something we can get a good observation of. Nice idea.
All right, so let's do one more round of applause.
This has been a production of Cornell University, on the web at cornell.edu.